and GCP or regulatory compliance. And again, with regard to monitoring planning under a risk-based monitoring paradigm, there are 3 key steps towards guaranteeing of quality. Those are protocol development, which drives directly and determines the specific nature and structure of the CRS. Consideration, that is to say, consideration of multiple potential factors, both intrinsic to the protocol but clearly also extrinsic, for example, site and patient factors. And finally, bringing all the input and assessments to bear on the development of an efficient and effective approach to monitoring of the investigation. So let's look at what can generally be considered as the critical to quality factors that need to be taken into consideration and thought about in developing a risk-based monitoring plan. So the indication being studied may well determine the risk to patients in the trial and thus the risk to quality as I defined it earlier. For example, certain specific indications within dermatology may involve lesser patient risks and certainly different ones than, say, a trial in acute coronary syndrome or transplantation medicine. There may be well-known risks characteristic of the investigational agent itself or its administration method if we're talking about interventional studies. Protocol complexity clearly determines risks to study integrity. And irrespective of the disease entity being studied, a particular patient population targeted may alter overall risks or the acceptability of those risks and thus the approach to monitoring. A couple of case studies. Here we have a phase 2 oncology study where the investigational product is with a new class about which very little is known from a broad safety profile. So for such a study, we would approach this with 100% source data verification of the first patient